All right, so we are here to talk about some of the foundational learning targets for photosynthesis. So I just kind of clumped together the first 1, 2, 3, 4, 5, 6, 7, because those all kind of came from that first Nearpod or that first presentation where we were talking about all the things that you need to know to kind of understand photosynthesis. And so the very first one is I can explain how producers obtain food or energy, because food is energy. So when we talk about food, we're really just talking about energy. And that is that they produce it themselves. That's why they're called producers, or sometimes you'll hear them called autotrophs. Okay, auto means self, troph means nourishment. You and I are heterotrophs, meaning other nourishment, because we have to eat things. But autotrophs don't have to eat. They're able to produce food or store that energy as food in their bodies. They're going to do this through one of two ways. They're either going to be photoautotrophs or they're going to be chemoautotrophs or chemiautotrophs. So it just depends on if they do photosynthesis, where they use light to synthesize glucose, or if they do chemosynthesis, which is where they use chemicals to synthesize glucose. So they break a bond, it releases energy, and they use that energy to make glucose. And so that's how producers obtain food or energy, is they use their environment, either other chemicals or sunlight, to produce glucose in their own bodies. Okay, the second one, I can explain how the structure of the chloroplast supports its function. So here is a chloroplast. These are the thylakoids. I don't know what that word means, but it's an interesting word. Out here is the stroma. Stroma is just a jelly-like substance, kind of a gel-like substance, and that is where cycles are always going to take place. If you'll remember in respiration, the citric acid cycle took place in the matrix of the mitochondria, which is, again, that fluidy gel-like filled area. And the Calvin cycle are dark reactions or light-independent reactions, all these different names for the same thing. They take place out in the stroma. All right, when you look at a thylakoid zoomed in, it is actually a double-membraned structure. So when we look at the thylakoid membrane, what this is it is a phospholipid bilayer. I shouldn't say a double membrane, a double layer membrane. So it is a phospholipid bilayer, just like you would see on a cell. It's the same kind of membrane right here too. So that's what it would look like zoomed in. All right, so how does the structure of the chloroplast support its function? Well, the stroma provides that oozy, I don't know, that's not a good word, but that kind of gel or jelly-like environment, which is what cycles need to take place. So it provides that optimum environment there. And then the thylakoid membrane, we need to do the light reactions, we need to create a concentration gradient. Okay, we're going to do chemiosmosis, and so we use an electron transport chain to pump hydrogens in. Okay, and it's going to create this concentration gradient in here, this high concentration, and those hydrogens they're going to ooze back out. And when they do that, it's going to happen through ATP synthase, and it's going to phosphorylate ADP to ATP. Well, in order for that to work, you have to be able to create a high concentration. And so what we do is we create this high concentration of hydrogens inside here. But in order for you to have a concentration gradient, there has to be a barrier. In other words, if I just had molecules, let's say hydrogen ions, just floating around, and some of them started moving over here, that's not a concentration gradient. This is not, yes, this is high concentration, yes, this is low, but in order to be a gradient, there has to be a barrier, a membrane that prevents them from just flowing freely. And so the fact that the thylakoid has this membrane, this phospholipid bilayer, is what enables us to create that concentration gradient, which is what enables us to create our ATP. And so structure goes with function. It is set up perfectly just for that. Another really cool thing is that the ATP, if you'll notice, is produced on the outside of the thylakoid. We're also going to produce some NADPH, again, on the outside of the thylakoid, out in the stroma. Those are the two things that we need for the Calvin cycle, or the light-independent reactions. So those two things are already going to be out in the stroma where they're needed. So again, another way that structure and function are linked. The next one is I can explain the link between the structure of a leaf and its function. So the leaf has an upper epidermis. It is covered usually by a waxy cuticle, which is protective. It also prevents evaporation because plants need water to do photosynthesis. So that cuticle, I don't have it shown real well here, but let's see, I'll do it in a highlighter. So you'll get the idea that it's kind of transparent. So there's that waxy cuticle. And that waxy cuticle is going to let light through, but not water. And so it's going to preserve water for the plant so that it has the water for photosynthesis, but it does allow light to come through because the plant obviously needs light. These up here are mesophyll cells. All of these up here, they're called palisade mesophyll. These are your spongy mesophyll. Mesophyll cells are the photosynthesizers. They are the worker bees when it comes to photosynthesis. So all of these mesophylls are going to be filled to the brim with lots, and I don't have room, and my pen's not giving me sharp enough point to show you, but I can't remember. I feel like I read something. I think it might have been in the Nearpod that I created, where it talked about the number of chloroplasts. It was something like 500,000 chloroplasts per square millimeter of leaf or square centimeter of leaf or something. It was ridiculous amounts of chloroplasts. And they are, for the most part, concentrated in this mesophyll. So the mesophyll is situated where it can get light. So some light's going to go through, some light's going to be reflected back out, and some light is going to, I should say, be absorbed. So sunlight will be absorbed by here, some will be reflected back out to wherever, and some light will be transmitted through and come down to these cells down here. So all of these mesophyll cells are getting light. They're all getting water, and that water is coming from the vein. So I showed a vein. I don't know if I talked about this in class, but the vein has two parts. It has a xylem and a phloem. Xylem carries water from the roots up, and so this vein is going to be bringing water into the leaf. And if you pick up any leaf, pretty much any leaf, you can see the veins in the leaf. And so that's what those veins are doing. They're bringing water up, and so that water is going to osmose out to these different areas. And then the other thing it does is it has phloem, which carries food. Phloem is P-H-L-O-E-M, but it carries food. I just always say phloem sounds like an F, and so that's how I remember it carries food, which for the plants is sugar. And so when the plants make this sugar, they give it to the vein, and the vein takes it down to the roots and stores it there for the winter. So when winter comes and they can't do photosynthesis because there's not a lot of sunlight and the days are shorter, then they can pull that sugar back up from their roots and survive. So structures that are important, cuticle is protective, okay, but it lets light through. Mesophyll cells are situated where they can get plenty of light. They're also situated near a vein where they can get water, and they're giving their sugar back to, so we can show water coming out in blue, and we can show, let's say sugars are pinks, so sugars are going into the vein and going down to the roots. The other thing you'll notice, and this is actually going into the next I can statement, is I can describe the role of the stomata in photosynthesis. Now here is the stroma in the chloroplast. Don't confuse that with the stoma or stomata. Okay, so I think stoma is singular, stomata is plural. I always get it mixed up. So just know if you see stoma without the TA, or if you see stomata, they're the same thing. They are different than the stroma. That R is important, okay? So the stoma means mouth, and so the stoma is going to be these pores in the leaf, and what's going to happen is when these pores are open, they can open and close, CO2 is going to come in, and oxygen is going to leave, which is great for us because we love oxygen. And so that CO2 is going to come in, it's going to be just going all around these spongy mesophylls, giving them CO2 for the light-independent reactions. It's going to go up here to these mesophylls. And so the fact that we have this spongy mesophyll allows that CO2 to go through. If all of this was like this first row and it was just packed in layer after layer of cells, the CO2 wouldn't be able to get to the upper layers. So the fact that we have this spongy mesophyll with lots of air gives our CO2 a pathway to get to these top cells. Same thing with oxygen. All of these are producing oxygen, all of these cells with the light reactions, and so that oxygen is able to diffuse through and back out. So the role of the stomata is really gas exchange. It's bringing in CO2 and it is letting oxygen out. The next is I can define pigment. A pigment is simply a molecule that absorbs a particular wavelength of light. Different pigments absorb and reflect different molecule or different wavelengths, and so that is why pigments have color. Because a lot of times when you think of pigments, you think of art, or you think of dyeing clothes, or you might think of dyeing your hair, and all of those are using pigments. But what makes a purple pigment purple is the fact that it is rejecting violet rays of sunlight, but it's absorbing all the rest. And so the fact that it's violet, I should say, not purple, But the fact that it's violet is because it is not using violet light. So plants are green because they can't use green light. And that really kind of throws everybody's brains for a second. But plants cannot really use green light very well, or at least the main pigments of chlorophyll A and B cannot. And so that's why that green light is rejected back at you, and that's why plants look green. So pigment is simply something, a color, a molecule that absorbs a specific color of light. I can explain the role of NADP plus in photosynthesis. NADP plus. NADP plus is an electron carrier. Electron shuttle, electron Uber, whatever you want to call it. It is just like its cousin NAD plus that we talked about in respiration. It just has an extra phosphorus or phosphate group. I like that. I think it's beneficial because it helps us remember that the one that has the P is in photosynthesis that starts with a P. All right. Now, NADP plus is going to grab an oxygen and 2 electrons and become NADPH. And these electrons are going to come from the light reactions. They're going to get zapped with sunlight, so they're full of energy. So these have a lot of energy. Anytime you see electrons moving, generally you're seeing energy moving. And so this is a high energy molecule. And it's going to take that energy from the light reaction over to the dark reaction or the light independent reaction. But we'll talk more about that when we get to those notes. So that's the role, is it's just going to carry those electrons from point A to point B. And then the last one for this is I can describe the process of photophosphorylation. Now we're going to get into this more when you're actually working through the light dependent reactions. So you might want to watch that video as well. But photo means light, and phosphorylation is a fancy word that means to add a phosphate group to something. Generally, and in all of our cases for AP Biology, we are talking about adding a phosphate group to ADP to turn it into ATP. And so what photophosphorylation is, it is using the energy of sunlight to provide that energy, and it's indirect. And if you remember the light reactions, you remember you've got that electron transport chain, the hydrogens get pumped in, they fall back through ATP synthase, and that's what actually makes the ATP. So it's not that the light is directly given this, but indirectly through a couple of other processes, it is providing the energy to make ATP. So that's what photophosphorylation is. We talked about in respiration substrate level phosphorylation and oxidative phosphorylation. This is the third and final type of phosphorylation we will talk about. And that's it. I hope that helps. If you have any other questions, please let me know.